Nucleotide excision repair is an important general process. Cells use nucleotide excision repair to correct many different types of DNA damage, including UV-induced thymine dimers. Nucleotide excision repair is found in both eukaryotes and prokaryotes. But the molecular mechanism of nucleotide excision repair is best understood in prokaryotes. In this video, we will show the process of thymine dimer repair in E. coli. The nucleotide excision repair process uses a set of proteins called as UvrA, UvrB, UvrC, UvrD. UVR stands for ultraviolet light repair of pyrimidine dimers, but we have to remember that this system is also used to repair other types of DNA damage. This process begins with the loading of a protein trimer onto the DNA double helix. This trimer consists of two UvrA molecules and one UvrB molecule. This trimer moves along the DNA scanning for damage that distorts the double helix. When the trimer encounters DNA damage such as a thymine dimer, it stops. The UvrB protein remains at the damaged site while the two UvrA proteins are released. UvrC protein then binds to the site. UvrC protein then makes two cuts on the damaged strand. Both cuts are several bases away from the damaged site. UvrD, a helicase, then binds to the site. UvrD then separates the segment of damaged strand from the rest of the molecule. UvrB, UvrC, UvrD are then all released from the damaged region. This leaves a gap. DNA polymerase is then recruited to the DNA. DNA polymerase then fills this gap with the proper nucleotides. DNA polymerase then leaves the DNA with a nick. DNA ligase then binds to the nick. DNA ligase then seals the nick. DNA ligase then leaves the DNA. The end result is that the damaged DNA has been replaced with the correct bases. Please like, subscribe and share.